Well, I want to welcome everyone back to the Connect Science webinar sponsored by Dana Farber Harvard Cancer Center and the Leukemia Lymphoma Society. I'm David Weinstock, and it gives me great pleasure today to introduce Dr. Bradley Bernstein, who is a professor of pathology at Mass General Hospital and Harvard Medical School, and also a member of the Broad Institute. Brad is really at the forefront both of technology development for single cell sequencing and also studies of the 3D architecture of the cancer genome. It's a tremendous pleasure to have him here. A couple of reminders. Please feel free to post questions in the Q&A tab at the bottom of your Zoom screen. And that we are very fortunate to have Kathy Wu speaking tomorrow on elements of personalized cancer vaccines. I think thus far we can all agree the seminar series has been tremendously successful. We're averaging around 1,500 participants and then an additional 1,000 online views of the talks, which are being posted within a day or two afterwards. I think I made a mistake. I said Kathy Wu will be tomorrow. That's not right. Tomorrow we skip the seminar. We take Wednesdays off for our internal Dana Farber meeting. So Kathy will be on Thursday, and then Ben Ebert will be on Friday. So with that, Brad, thanks again for joining us, and please take it away.
All right. Well, thank you, David, for the nice introduction. I'm really honored to be giving the talk, although I wish I could be doing it in person with everyone. It sort of reminds me of a conversation I had some years ago with Mandy Fisher, who's a renowned British epigenetics researcher. I think Mandy was needling me a little bit about how much massive amounts of data that we Americans, and particularly we genomicists, tend to generate in the lab. And she explained to me that British scientists were very different. That the archetypical British scientist would head off to the library for 364 days of the year just to plan one experiment. But then on the 365th day, they'd head back to the lab and voila, a miraculous discovery. Now, I'm sure there was some hyperbole in her description, but I do hope you all find ways to advance your science in these difficult times and that your first experiments back in the lab are roaring successes. How do I move slides? Good, next slide. So onto the science of epigenetics. It's been nearly 15 years since I started my group at MGH in the Broad Institute studying chromatin epigenetics. And the field has just changed so remarkably over those years. You know, in 2005, we had a recently completed draft of the entire human genome, but we knew very little about how cells remembered their fates or, about how genes were controlled. And I can recall Tim Bester presenting this slide at a meeting about that time. And he sort of joked that if you have a known gene, but an unknown protein, of course, you were studying genetics. If you had an unknown gene, but you knew the protein, of course, you were doing biochemistry. But he joked that if you didn't know your gene, you didn't know your protein, this must be epigenetics. And I think this was Tim's way of protesting that, our field was a bit of a mess, and virtually any baffling biological process that couldn't be explained would be sort of chalked up to epigenetics. And I'm just seeing one person who can't hear the speaker. I just want to make sure, can other people hear me okay?
Yeah, we can hear you fine.
Okay, good. Just one person needs to turn on their volume, turn up your volume. So this is sort of where the field was when I was getting into it. And you remember the joke was, this must be epigenetics. So, but we've come such a long way in the past 15 years towards understanding the fundamental processes that control cell fate and identity, gene regulation. We've learned that there's about 20,000 protein coding genes in our genomes, yet these only comprise about 1% of the real estate of the human genome. We've learned that the other 99% is packed with regulatory elements that, like enhancers, that control which genes are read, where and when they're read. Of course, all this genomic DNA is packaged into proteins with along with RNAs into chromatin. We've learned that chromatin is this highly dynamic, has critical functions. Active genes are packaged into sort of these open states that give access to transcription factors and transcriptional machinery. Repressed genes are sequestered in sort of compact chromatin that silences them. And of course, these alternate states of chromatin, they're dynamic in development. So you have to switch in response to developmental cues or environment. So these switches then are controlled by these scores of epigenetic regulators that can methylate DNA, modify histones, remodel nucleosomes. So this emerging knowledge of chromatin and epigenetics is having such a big impact on the many fields, but in particular on cancer biology. And cancer epigenetics is, of course, the major focus of my lab. So cancer is typically often thought of as a genetic disease where mutations turn on oncogenes or inactivate tumor suppressors. Yet we know that changes in chromatin structure, methylation, can also turn on genes on and off. And in fact, there's a very strong case to be made that such epigenetic defects act very broadly in cancer. DNA methylation patterns are altered in most or all tumors. Chromatin regulators are very frequently mutated. They can be very early events. And epigenetic therapies are now in the clinic. Yet a challenge has been, for us to really understand what are the underlying mechanisms that are sort of by which these, epigenetic alterations impact tumorigenesis. And that's I think, that's been the central challenge. It's critical to understand this if we're going to effectively deploy all these epigenetic inhibitors and modulators in the clinic. And this is really what I'm going to talk about for my presentation today. I'll focus a lot on DNA methylation. So methylation is really, it's a bona fide epigenetic mark. And when I say that, I say that because methylation can be transmitted through cell division. Sorry, I'm moving around a little bit. Just like a genetic mutation, right? When the strands are copied, when cells go through mitosis, of course, you copy the sequence and you can copy any mutations. But there's also on the epigenetic side, shown here, you've got these maintenance methyltransferases, DNMT1 in particular, whose job it is to faithfully replicate the methylation patterns as the DNA is replicated and cells go through mitosis. By the same sort of concept, if a methylation pattern is changed in a cell, and I'll talk about how that can happen, that is heritable, mitotically heritable as those precancerous or cancerous cells divide, maintained. So it's like an epimutation. It's a lot like the genetic mutations that I think are more familiar to most of you. Now, albeit I would just say that, the methylation patterns are not copied quite as efficiently as the DNA mutations. And I think that's important for some of the things I'll talk about. All right, so genome-wide methylation patterns are well recognized by many colleagues in the field, and Steve Balin, Peter Jones, Andy Feinberg, Jean-Pierre Issa, Peter Laird, and colleagues, to show that these patterns, genome-wide patterns, change profoundly in cancer. There's two things that can happen at a very sort of gross level. You have these GC-rich promoters that tend to be unmethylated, have very low methylation in normal developing cells, but in cancer, they can become focally hyper-methylated. They gain methylation. At the same time, you have these deserts where you have very few CPGs, few genes. These are normally totally methylated, but in cancer, they become hypomethylated. We have some music in the background.
God, you look so pained. What's wrong?
We've got some noise in the background.
I'm going to try to find it and mute it. Hold on.
Okay. But even though these are sort of the best characterized patterns, changes in epigenetic changes in cancer, we still don't have a great idea about how they impact tumor biology. And what I'm going to do now is I'm going to share two recent stories from the lab that have uncovered really unanticipated consequences of these methylation changes, including sort of novel mechanisms that are, I think, challenging current views. And I'll talk about those. But really, to understand how this all works, we need to think about how the genome folds up in cancer cells. So I'm going to come back to methylation later, but I'm going to talk a little bit about the genome. In fact, comprises about 6 linear feet of DNA that must be packaged into a nucleus smaller than the sharp end of a pen. It's precisely folded in each cell such that exactly the right set of genes, enhancers, regulatory elements are made accessible so they can execute these exquisitely cell type specific expression programs. What could possibly go wrong with this, right? So for many years, my lab has been investigating genome topology with the goal to understand how these topological structures are altered in cancer and what their impact might be. So the first story I'm going to tell you today is about a large-scale sort of atlas survey study where we've taken a really unbiased look at topology in colon tumors. And second, I'll describe a more directed study where we've identified specific alterations that in GI stromal tumors that activate oncogenes. So these are tumor-driving epigenetic events, no change in genomic sequence. So I'm excited about these projects. I think they kind of teach us something fundamental about epigenetics and genome organization. But as you'll see, they also have very specific translational, they bring very specific translational opportunities too. Okay, so morphologically, irregular or atypical nuclei, as I'm showing here, are a pathological hallmark of malignancy. This histological feature is, of course, critical for accurate diagnosis of cancer, prognosis, yet its molecular underpinnings are not well understood. So this has really perked our interest in understanding how the genome folds in cancer. And at the same time, the field of genome biology has increasingly turned its attention to the 3D genome. We've learned that genome organization is hierarchical. You've got these multiple layers here. You have DNA methylation and histone modifications and remodeling, and that sort of act to sort of package individual loci into open or closed states. You then, in sort of euchromatic and heterochromatic states, you have chromatin loops that can be formed between enhancers and promoters. This is on the order of perhaps tens of kilobases or 100 kilobase. You then have these TADs, which I'll talk a lot about, topologically associating domains, which are on the order of hundreds of kilobases to megabase in size. So TADs are these, regions that have frequent internal or intra-domain interactions, but TADs tend to be insulated by flanking boundaries. And finally, the TADs themselves are segregated into 2 large and spatially separate compartments. Compartment A, which consists of gene-rich, open, and accessible chromatin, and compartment B, which is gene-poor, closed, and more compact. So you're probably familiar with how we measure DNA methylation and by sulfite sequencing and histone modifications by chip sequencing. But I'll take a moment to explain how these higher ordered organizational levels can be studied. So a primary strategy is these proximity ligation methods shown here. Cells or tissues are fixed and the nuclei are isolated, and the genome is cut up with restriction enzymes. So after it's cut, these cut DNA molecules still in situ in their native folded context are religated. The idea is that sequences that are very close in 3-dimensional space, even if they're not close in linear DNA space, if they're close in 3-dimensional space, they're more likely to ligate to one another. And in a sense, this ligation frequency is a measure of sort of 3D contact or frequency of contact in the nucleus. Now we can do this in an all-by-all manner by sequencing everything in high C, as I'll show you. We can also target specific loci by 4C, as I'll touch on as well. Now, colon cancer is really an established model for cancer epigenetics with characteristic changes to DNA methylation and chromatin. But very little is known about genome topology in colorectal tumors. So this prompted Sarah Johnstone, a very talented postdoc in the lab, to characterize topology of a large cohort in this, and she's done more than this, but this was sort of the initial STAB, collecting Hi-C and other data to map out the topology of normal colon tissues and colon tumors. And I think her work provides one of maybe the first sort of comprehensive view of genome topology in a solid tumor. And it's really changing our thinking in a significant way, as I'll tell you. So one of the most prominent topological features in these maps, these Hi-C maps, is, as I told you, is these TADs, are topologically associated domains. These are insulated neighborhoods of genome that self-interact, but are partitioned from adjacent genomic regions by boundaries. These are sort of the functional unit of your genome. I'll tell you why. You know, you can conceptualize a TAD actually as an extruded loop, pretty large loop of hundreds of kilobases. An enhancer in a given TAD or given loop here can interact with genes in the same TAD, but it can't contact genes in a neighboring TAD. So this is why, you know, CTCF sites are sometimes thought of as insulators. This is why we think of these as sort of insulated neighborhoods, these TADs. These are the functional units where all that activity between those many enhancers in the genome and those many genes are constrained, okay? So TADs can be appreciated in the Hi-C maps. So this is Hi-C map data. Basically, it's a pairwise interaction map. The x-axis here is chromosome 7. The y-axis is also chromosome 7. And the red heat tells you about interaction frequency between 2 points on chromosome 7. You see there's a nice red diagonal because points that are linearly very close along the chromosome interact a lot. But if you zoom in, this little blow up here on the diagonal, you can see that there's these little boxes of high self-interaction. So you can, here I rotate them a little bit just to make them clearer here, these little triangles. So these are the TAD regions. This is the TAD, lots of interactions, and then in between them are the TAD boundaries. And these TAD boundaries are remarkably enriched for the CTCF. They bind, this is where the CTCF insulator protein very frequently binds. All right, so we've looked at a lot of tumor types and samples. What we find is that TAD boundaries, the TAD structures themselves, are extremely stable in normal colons, tumors, cell lines, and other tumor types, okay? Most of them are preserved. All of them are preserved. The exception is hypermethylated tumors. This subset of tumors that gain methylation to a profound degree are an exception. And that's so that actually is going to be the topic of part 2 of my talk. But first, I want to move on to sort of the next layer of organization, and that is this long-range compartmental organization changes in cancer. So another really interesting feature of the Hi-C maps are these sort of off-diagonal interactions that make these maps kind of look like a plaid shirt. If you look at the patterns very carefully, I think you can appreciate the different compartments, right? So this is a TAD here, and it makes interactions with TAD here and here and here and here. And what's common about all those TADs is that they're all gene-rich, kind of active. They're all compartment A. They come together spatially to a degree in the nucleus. And that's, in contrast, here's another TAD right here in the red, this box here. You can see it kind of makes inverse long-range interactions with sort of the opposite kind of regions. That's because this is a compact inactive region. So in fact, if you process Hi-C data with an eigenvector analysis, the first component separates the whole genome into 2 compartments, a gene-rich compartment A and a gene-poor compartment B. Okay. So what we wanted to do then is ask, you know, how do compartments, how are they organized in normal and tumor, and do they change? And basically we did a differential analysis I'll show here. And here you, basically what I'm doing is we're taking a region, showing a region just like a Hi-C map, except it's a differential Hi-C map. So red heat tells you about interactions that are gained in tumors, and green are interactions that are reduced or lost in tumors. And what hopefully you can appreciate is these red heat always corresponds to an interaction gain between B and A. Basically tells you that there's more intercompartmental interactions between these compartments in tumors. It's essentially the normal compartmentalization or the normal compartmental partitioning is sort of breaking down in the tumors. And really, to emphasize, this didn't come out of cell lines because these patterns are really distorted in cell lines. The only way we arrived at this was really to directly compare human specimens that were either normal colon or colon tumors. Okay, so this is high C data. But you might ask, okay, these interactions and compartments are a little bit abstract. What do they really look like? You might know that chromatin in the nucleus is sort of asymmetrically distributed. Inactive lamin-bound parts of the genome tend to be located peripherally, shown here. Here's sort of these lamin-associated TADs. They're very inactive. They're outside. And then inside, are these active regions. They're more interior. Of course, it's all averaged out a little bit. It's very dynamic, but this is a general picture. So in order to sort of understand this in our data, we teamed up with Ben Zhang, who's a polymer chemist, computer scientist at MIT. And what Ben did was he simulated models of how the whole genome folds up very rapidly, and he iterated them. And he iterated these models again and again until they match our high C data. And if you think about it, you can make a model in a computer, and then you can essentially estimate what that fold up model would look like if you did a high C experiment. And that's what he did. And he found basically built in silico models that corresponded to normals and tumors. So we're very happy to see that his model for the normal colon nucleus positioned B, the inactive region, sort of on the outside, and A, more interior. And we're struck that his model for the colon tumor nucleus really showed that this sort of disorganization, this loss of partitioning, seemed to be accompanied by sort of a disorganization of where these compartments are positioned in the nucleus. Okay. Major spatial reorganization in tumor nuclei. Okay, so of course, naturally, we see these models. We want to visualize them spatially. So one technique we use is electron microscopy, where heterochromatin stains is very dense, and it stains darkly, whereas euchromatin stains lightly. This is actually a sort of normal colonic epithelial cell. You can see the dark staining around the outside. That's the heterochromatin. On the inside, it's sort of fluffy chromatin. This is the euchromatin. There's the nucleolus. Okay, so what happens if we look at sort of normal colon nuclei? Here's another picture similar to the one I just showed you. You can kind of, imagine this is the polymer model from the high C data. It's not so far off, right? It's positioning things in generally the right place. But what if you do an EM of colon tumor nuclei? We're really struck by sort of this loss, this disorganization, and you can see that the heterochromatin is starting to move inside, and it's losing its tethering to the lamin. In the tumors, I think very consistent with what we're seeing based on the polymer modeling. So this was nice validation of the high C modeling data. Okay, so now we want to relate the changes in compartment structure. Remember, I talked earlier a lot about methylation. So now I want to try to tie these pieces together and relate the changes in compartment structure to methylation changes. And one of the most profound methylation change that happens in almost all tumors is this hypomethylation of large blocks. This is work from Feinberg and Laird and others. These large blocks become hypomethylated. So this is like a 4 megabase region. Normal colon has a nice methylation level, but the colon tumors reduced across this whole region. What we were really struck is whenever we saw these blocks of hypomethylation, they always corresponded to a TAD or several consecutive TADs. So there's a nice correspondence between where these happen. But there's more than that, because if you look at what types of compartments become hypomethylated, it was very clear. Compartment B, essentially all of compartment B becomes hypomethylated, which was, I guess, not so surprising, a little bit about these are inactive regions. But what was striking is that there were also these regions in compartment A, originally assigned to compartment A and normal, that seemed to be switching towards B, so becoming more compact in the tumors. And look, these regions correspond beautifully with the hypomethylated blocks. In other words, what it's looking like to us is that you kind of have another compartment. You have another region that used to be assigned to A, in a simple assigned to A, but looks different, and it seems to be it seems to be intermediate and changing its structure in tumors, and it corresponds beautifully to hypomethylated blocks. Okay, so if you have this close association with hypomethylated blocks, it allowed us to do something. We could now take all of the regions assigned to compartment A, and we could find all the ones that were corresponding to hypomethylated blocks. We could say these must be, these may be a new compartment. We'll call it compartment I because they seem to have intermediate eigenvectors or intermediate properties. Okay. So if we look at the modeling, Binzhang's modeling data, you can see again that, you know, B is on the outside, A is on the inside, and I seems to be sitting at the interface in the models. And I'll show you one other piece, and that is that I not just distinguished by its positioning, it's distinguished by its chromatin state. So remember that these are the different marks. These are the different compartments you can see here. B is getting hypomethylated, and it's gaining a lot of K9Me3. In dark blue is A, and it's acetylated and active. But look at light blue. Light blue, formerly assigned to A, now we're going to call it compartment I. It is gaining K27Me3. So this is the conditional, more of a conditional heterochromatin mark. So you've got this switching into compartment I. So we think that, you know, because of this alternate methylation state, this alternate topological positioning, and the alternate chromatin state really argues that you should explain this by a 3-compartment model of A, I, and B, really important in tumors. And it's a pretty significant rework because compartment A actually corresponds to about 10% of the genome. Okay. Now, Carmen Adrians, who's a super talented postdoc in the lab, she jumped in and she helped us to visualize compartment I by FISH because, you know, a picture is worth 1,000 words. What she did is here she's doing HCT116 colon cancer cells. We now are generating and analyzing actually data for colon tissue and ultimately for tumors. And we see similar things, although we haven't done all the analysis. It's looking like this should hold. But let me tell you what we found in the HCT116. You can see that we've done FISH with, you know, thousands of FISH probes. They're labeled with different fluorescent dyes. So you can distinguish the B compartment, which I'm labeling here in yellow, the A compartment in dark blue, and the I compartment in light blue. And so this is a beautiful example of the B on the outside, I is intermediate, and A is most interior. Of course, this is one cell, but Carmen has now analyzed, I think, about 300 cells and looked at the distribution of each of the compartments. And you can clearly see that the B compartment in yellow is the most peripheral. It's at the edge of the nuclei. A is the most interior, and I is intermediate. So this is imaging data that exactly confirms what I've just been telling you from the Hi-C data. Okay, so what are the causes and effects of these compartmental shifts? First, I want to tell you, in terms of causality, we think that the hypomethylation actually probably plays a causal role in all of this. And I'll tell you a couple of reasons. One is there's a very close correlation between the extent of hypomethylation and the more something is shifting over to be B-like in the tumors. So this is, you know, of course, a correlation, but look, the more hypomethylated you are, the more B-like you are. Compartments I start A, they get hypomethylated, they shift over to B, they become more compact. Still, it's a correlation. But Sarah went on to treat some HCT116 cells with DNA demethylating agent to reduce their methylation levels. And what this did is it gave rise to a number of new hypomethylated TADs. And what we were struck by is when you demethylate and you hypomethylate a TAD, it tends to shift towards B. So this is really suggesting that all this shifting we're seeing that correlates with methylation may well be driven by the hypomethylation itself. And I want to show you one more result on this slide that I think is very important. One might have guessed initially that the loss of methylation might, in compartments B and I, might de-repress genes, because, you know, methylation is thought of as repressive. But that's exactly the opposite of what we found. What we found instead was that these compartments that become hypomethylated actually get repressed, okay? The genes, the few genes, not a lot of genes, but the genes that are in there, they have lower expression in tumors. So let me summarize what I've told you so far. In normal colon tissues, you've got compartments A, B, and I, and they're really nicely partitioned from each other, and they're spatially segregated in the nucleus. But this organization is severely compromised in tumors. Compartments B and I both become hypomethylated. I is gaining this repressive H3K27 methylation, and B is getting even more K9ME3, falling away from the lamin. We think, though, their repressive chromatin states are actually doing just a fine job of sustaining their compact and repressive character, because the overall transcriptional output of these compartments is actually reduced in tumors. Okay, so the tumor biologists in the audience who are listening today are probably now asking, you know, how does this all impact tumor biology? What's the relevance? And we were asking ourselves exactly the same thing. Our first hypothesis was sort of a naive hypothesis, one that you might have thought about 5 years ago in the field, and that is that, you know, maybe the hypomethylation is de-repressing a bunch of oncogenes in these compartments, and they're coming on and they're driving tumors. But as I told you, most genes are going down, and this is not the case. Hypothesis 2 was that maybe the compaction of the compartments is downregulating tumor suppressors, in that way driving tumors. But we looked, and this is not the case either. So we kind of reject our two first hypotheses. Maybe we're not thinking about this in the right way. So let's step back. You know, block hypomethylation was initially described in cancer, but more recently it's been recognized to be a common feature of cells that have accumulated way too many cell divisions. So I'm just showing an analysis of aging fibroblasts in culture, and it's showing that compartments B and I, not A, but compartments B and I become increasingly hypomethylated with days in passage. That is, the more times the fibroblasts divide, the more hypomethylation you get. We also borrowed some data from a recent publication, sort of a population epigenomic study from Grady and colleagues, where they actually biopsied about 300 normal colons of people of different ages. And the nice thing is you can see, again, this nice correlation as individuals age. You don't see it quite as nicely in B, but clearly in I, you see a similar phenomenon that as individuals age, their colon becomes progressively hypomethylated. We think as the cells divide, but we'll get into that a little more. Okay. I can also tell you, remember what started all this discussion was this compartmental disorganization I showed you here in tumors. You can see this disorganization, this aberrant interactions between compartments A and B. You can see it in tumors in this plot here, just a quantitative single value, purple or tumor. I can tell you, you also see it in fibroblasts that have been passaged and passaged and passaged to replicative senescence. So again, cells that are divided too many times, they get the same phenomenon. And we're actually started to think that maybe this fundamental compartmental shift is actually reflects some kind of epigenetic response to excess cell divisions. So let's look at the gene expression changes themselves. So what actually happens? So block hypomethylation is a good surrogate for compartmental shift. So we could use methylation patterns to begin to ask what happens to gene expression. Now the TCGA has published methylation expression data for about 150 tumors. So we were able to use those genes to identify genes in compartments B and I whose expression correlates with hypomethylation. So basically, each of these dots in the picture here is a different gene, and it's plotted in a volcano plot by whether its expression increases the more a tumor gets hypomethylated, or whether its expression is reduced as a tumor gets increasingly hypomethylated. I'll tell you that, most of these genes on the right, actually, in green and red, Well, they're cancer germline antigens in red and ERVs. And kind of we knew that that loss of methylation de-represses some of these markers. And they're associated with anti-tumor immunity, which is really interesting. But the vast majority of genes are being repressed, as I told you, in association with hypomethylation, and we think compartment change. So we took these genes and we asked, what kinds of genes are they? What we found is that these gene sets, if you put them into Go analysis, they're enriched for programs for stemness programs. They're enriched for EMT, invasion, metastasis. In other words, the compartmental change that we think arises because cells have divided. As cells divide too many times, does a couple things. One is it turns on tumor immunity genes that might cause, a cell, a wayward cell to be immunologically pruned. And the second thing it turns off stemness and metastasis programs, which seems like a pretty good adaptive response to maybe a cell that's acquired a lot of cell divisions and might be at risk for acquiring mutations and becoming cancerous. So this is starting us to think that really this is all a tumor suppressive process, which makes a lot of sense to us, given that it's already seen in these other cell types that have acquired so many cell divisions. Clearly, the tumors have to overcome something, and through genetics and so on. But we think that these changes are suppressive. Let me give you one more piece of data, and this actually just very recently, this paper came out. I touched up on it a minute ago from Grady and colleagues, where they biopsied like 300 and something normal colons. Now, I didn't tell you before I just showed you data for all the normals. But it turns out of the 334, there was some number of individuals that actually had concurrent colon tumors somewhere else in the colon. So you can, they didn't sample those parts of the colon. They sampled a distinct part of the colon, normal colon. But you can think of those individuals as sort of high risk for getting colon tumors. And we were really struck when you look at the average methylation of the ones that are low risk, aren't getting colon tumors, is here, it's low. But the average methylation in your colon of those that are at high risk are getting colon tumors is much higher. So really consistent with the concept that maybe this progressive hypomethylation that occurs with aging is protective, and it's preventing tumor progression. Okay, so I want to just sort of summarize this part of the talk before I move on. So basically, I've shown you a comparative study of genome topology in normal colon and colorectal tumors, and really highlighting sort of profound changes in compartmental reorganization. Compartments A, B, and this newly compartment I, nicely partitioned and asymmetrically distributed in the nuclei of normal cells. But in tumors, this is lost. And the inactive B and I compartments, B falls away from the periphery, from the lamin. It's actually more compacted by repressive chromatin modifications. And this enacts transcriptional changes that we think are inducing tumor immunity. We think maybe repressing stemness and metastasis. And we think overall helping to reduce cancer risk. This is an impediment that tumors must overcome. Okay. I'll briefly summarize this at the end, but I'm going to now move on to, as promised, part 2. So what I've just told you about is sort of an epigenetic and topological mechanism that is suppressive or suppresses tumorigenesis, okay? But now I want to talk about another kind of topological change that's actually very clearly doing just the opposite. This is a distinct kind of topological changes that is driving tumorigenesis by turning on oncogenes, okay? It's related to these topologically associated domains, or TADs, that I mentioned earlier. Now recall that TADs, are these, the boundaries are very stable except for in hypermethylated tumors. They sit here and they constrain enhancers to only act on genes that are nearby. And I told you that their boundaries are enforced by CTCF. What I didn't tell you is that CTCF is a methylation sensitive binder. When DNA gets hypermethylated, the CTCF motif gets methylated. CTCF can no longer bind to its binding sites, okay? All right, so I'm going to focus on GIST, and we honed in on GIST for some reasons that you'll see in a second, really for understanding based on the genetics or lack of genetics in some of GIST tumors. So it's the most common GI tract sarcoma. The GISTs are typically caused by these activating gain-of-function mutations that turn on the KIT oncogene. 75% of cases, and these tumors famously respond to imatinib. However, there's a subset of GISTs, about 15% of them, that don't respond to imatinib. They actually don't have any KIT mutations. The KIT locus is wild type. There's no other mutations that can be identified, except they've lost the SDH, which is this metabolic complex in the mitochondria. They're SDH deficient. Okay, and it's known that when you lose the SDH, this leads to hypermethylation of the DNA. The reason is SDH loss leads to too much succinate, which blocks the demethylases, and over time the balance shifts to hypermethylation. All right, so Will Flavahan, terrific postdoc in the lab, teamed up with Yotam Dreyer, who was a postdoc in the lab at Hebrew University, and we collaborated with George Dimitri and Jason Hornick and Matt Hemming at Dana Farber and others to sort of put together a cohort of GIST tumors of different subtypes, both KIT mutant and SDH deficient. We looked at normal stomachs and we mapped topology extensively. What we found is that SDH deficient GISTs, the hypermethylated ones, are losing thousands of CTCF binding sites, and as I'll show you, as a consequence, their genome topology is really changing. Okay. So this kind of goes back to there's a challenge in epigenetics that if you look at a tumor, a lot of things have changed in a tumor. There's a lot of epigenetic changes in tumors. How do you sift through all those changes and identify the subset that might be drivers, right? Drivers from passengers. So here's a stab at that. So what we've done is we've taken all the CTCF sites in the genome. Each dot is a CTCF site, and this tells you that the ones on the left of the volcano plot, these are the ones that are being lost in SDH deficient GISTs. These are the ones being gained, and you'll see not many of them are gained, very few are gained. Really what you have is a wholesale loss of CTCF binding events. And I colored some of the dots red. The dots in red happen to be the ones that get methylated in the SDH deficient GIST. So it really fits together that this hypermethylation is displacing a whole lot of CTCF binding sites. Okay. So here's a sort of another picture of the TAD, and this is kind of what we're looking for. There's a lot of TADs contain oncogenes. So oncogenes tend to be sort of isolated because you want to regulate them very carefully. They drive growth. If you look through the genome, you'll see that a lot of times if you have an oncogene, it's kind of isolated in its own TAD or maybe with one other gene or something like that. What we're looking for would be cases when you have an oncogene in a TAD where there's like a great big enhancer, in the neighboring TAD. We were reasoning that maybe if the CTCF site that was separating the two of them was lost, you could refold this into one great big TAD and the enhancer might turn on the oncogene. So we looked sort of systematically for super enhancers, and we looked for deregulated genes. We found that a couple things jumped out at us. One was the KIT oncogene, which is very interesting, but I'm not going to talk about just because of time. And the other was through the FGF locus, the FGF oncogene, which is quite fascinating, and I am going to talk about. I'll point out that the FGF oncogenes are massively induced, specifically in the SDH deficient GIST subtype. All right, so now here is a sort of a map of the FGF locus. You can see the FGF3 and the FGF4 genes. These are oncogenes. They drive FGF signaling. You can see this little triangle here tells you that this is a TAD, and there's a nice TAD that keeps them safe, right? It turns out that the neighboring TAD has a massive superenhancer in GIST, probably a massive superenhancer also in the cell of origin. And the reason why you need this superenhancer, it seems to turn on this gene called ANO1 that's also known as DOG1 or discovered on GIST. This gene is like the biomarker that the pathologists use to diagnose this tumor. It's really highly expressed. It just happens to be in the neighboring TAD, right? But look what happens in these hypermethylated tumors. There are these nice CTCF sites here. This is a track showing the CTCF sites. In the SDH, this is KIT mutant, but in the SDH deficient hypermethylated tumors, these CTCF sites are getting methylated and disrupted. You lose CTCF binding, okay? So now, you know, what I was telling you is, are we now taking this whole region and making this into one TAD and allowing the super enhancer to crank on these oncogenes? Let's take a look. Okay, we're going to use this technique 4C-seq because it allows you to start from one site in the genome and ask who it's interacting with. So if you take these are KIT mutant GISTs on the top, they're SDH intact, so they're not hypermethylated. Here's that insulator that's protecting FGF. Here's the super enhancer. The super enhancer, this is your viewpoint, and basically what this black tells you, you got a lot of interactions. The super enhancer is interacting with itself. And as soon as the super enhancer, as soon as it reaches that FGF insulator, that boundary, the boundary of the TAD really blocks it, and you don't get much bleed through, right? And you don't get any interaction with FGF3 and 4. But the situation's totally different at the SDH deficient GIST. Remember, I told you that the CTCF sites are all gone from this insulator. So now this ANO1 anchor point, there's massive interaction, bleeds past the boundary right into the FGF3 and FGF4, lots of interactions with the genes. We think this explains the very high expression of these genes in the tumors, and I'll show you some functional data to support that. We went back to the GIST T1 cell line that is, it's a KIT mutant. So remember, it's not hypermethylated, and the CTCF insulator is intact. These genes are off. But what we did, Will Flavahen edited the CTCF motifs and got rid of it. See, you lost CTCF binding, shown here, and look what happens to the expression of these oncogenes. This FGF4 goes up 6-fold, and FGF3 goes up like 35-fold by RT-PCR. So when you lose this insulator, you're cranking on the genes. And mind you, this insulator is like 100 and something kilobases away. All you did was you changed a couple bases, so CTCF couldn't bind. You didn't muck around with the gene, anything near the gene. This is far away, and so these are really long-range interactions, but they're potent long-range interactions, and they really suggest that in the tumor, that loss of CTCF is what's driving the expression. If you think about this as an epimutation and you draw an analogy to mutations, you want to think, is this a driver epimutation, like a driver mutation? A driver mutation should be very specific and it should be recurrent and specific to that context. So let's look at the methylation. The methylation of the FGF insulator is seen in every SDH deficient GIST that we've looked at. And I'll tell you, we have never seen it in any other cell type. It's not in any of the other types of GISTs. It's not in normal stomach muscle. We pulled down all the different tissue types in ENCODE. None of them have any methylation of this insulator. This is a constitutive insulator. It's always intact. The only time we've seen it lost is in SDH deficient GISTs. All right. So then I've really pushed on this, the idea that these insulator events are propagated through a clone, right? So, you've got CTCF. If you methylate the site, this is, if people think about parent of origin imprinting, this is how parent of origin imprinting, like IGF2, parent allele specific expression of IGF2 is maintained because one allele has CTCF bound and the other allele is methylated. This is really stable. Okay, it's an epigenetic switch. You didn't change sequence at all. What you did is you methylated the CTCF site and CTCF was displaced. So this is just like a genetic mutation. So we can borrow the models from the geneticists, right? We can say, okay, so let's imagine we have some progenitor cell. It gets this SDH lesion. It starts getting hypermethylated. You get some kind of epigenetic plasticity. So methylation starts displacing different CTCF insulators. You make epiclones, right? Because it's heritable. In most cases, they're passengers. In most cases, the loss of CTCF doesn't really have any consequence. It might or might not regulate a gene, but it doesn't regulate an oncogene. But eventually, you'll get this driver event. This driver event being methylation of the insulator that is protecting FGF4. FGF4 expression is cranked on, and the cell, some pre-tumor cell gets a nice fitness advantage and expands, and this ultimately gives rise to the tumor. Okay. So I want to move on from sort of theory to clinical application, though. I know a lot of people are interested in, what's the impact here. So, you know, the SDH deficient GIST, they don't have any oncogenic RTK mutations. But what I'm telling you is they're cranking on FGF, so they're probably, I'm arguing they're going to drive RTK signaling, right? And indeed, if you look at expression programs for MAPK signatures, you can see that the SDH deficient subset has very high MAPK signature, a lot like the KITs. So they are turning on signaling. I can tell you also that the SDH deficient GISTs have high KIT expression. And importantly, they have high FGF receptor expression. So I told you that FGF's getting cranked on, but the receptor's also there, right? So you've got, autocrine signal, potential for autocrine signaling, right? And here's the expression of FGF3 and 4. Okay, so if this is the case, what's the therapeutic implication? So we teamed up with George Dimitri and colleagues and Praful, and we were able to establish a PDX model of SDH deficient GIST in a mouse. We've validated that, you know, this model, the xenograft, had an RNA expression pattern that was almost identical to the parental tumor. It had methylation patterns that were identical to the parental tumor, and it had sort of insulator patterns. So this was a really faithful PDX model that was, replicated the primary tumor itself. So we went ahead and treated the PDX. We treated the mice with either a KIT inhibitor. You can see here for 28 days of KIT inhibitor in green. Doesn't do a whole lot. This won't be so surprising to the clinicians because it's generally known that this subtype of GIST is not sensitive to the standard of care KIT inhibitors. This is why we needed a new drug for this. We then treated with FGFR inhibitor, and I think you can see very strikingly throughout the treatment period, the 28-day treatment period, totally suppressed tumor growth. And then only much later did the tumor sort of grow out. And combination therapy may be even more effective. So I guess I want to just sort of summarize and sort of step back and think about sort of some of the big picture of some of this work and some of the parallels. So I showed you, I think there's really striking parallels between this epigenetic mechanism of oncogene activation that I've shown you and classical genetics, right? In GIST, you typically have a KIT mutation that activates oncogenes. It's genetic. But what I hope I've convinced you is that that in this weird subtype, SDH deficient GIST, you actually have an epigenetic lesion that cranks on oncogenic RTK signaling through FGF and KIT. What I didn't tell you about is a couple of years ago, we had found a similar phenomenon in IDH mutant gliomas. Now, involving PDGFRA. Now, PDGFRA is often genetically amplified. This is your oncogene activation, is genetic. But in IDH mutant gliomas, the PDGFRA locus is wild type. What we found instead is that there's an insulator loss, just like I showed you in GIST, but in the PDGFRA insulator locus instead is cranking on PDGFRA expression, and we think helping drive the IDH mutant gliomas. In both cases, I can tell you that clinical trials are sort of underway to test these hypotheses with George Dimitri and colleagues looking at FGFR inhibitors in the GIST and at MGH with Isabella Arlaga-Romani looking at whether demethylating agents in IDH mutant glioma can kind of restore the normal locus topology and help these patients. I also want to sort of step back and just remind you about the beginning of the talk, and that was we think we've identified an epigenetic mechanism of tumor suppression, right? Where remember that compartmental reorganization is suppressing these stemness programs in aging cells, and we think sort of forming a block to prevent tumor genesis, tumor progression. And I would just say here, I think this is an epigenetic and analogous, very analogous to a genetic mechanism. Again, it's kind of analogous to telomere shortening, if that helps you sort of get an impression of what we think is going on here with the mechanisms. All right, so I just want to wrap up. I want to give special thanks to Sarah Johnstone, who led, she's a superb physician scientist. She led the colon cancer work. I want to also thank Martin Ari, faculty in MGH pathology, and his postdoc Alejandro Reyes. We generated petabytes of data, or maybe more. I don't know. Martin could clarify, but they've been just incredible at helping us with the analysis of this massive data set. Carmen Adrians, I mentioned, has done the imaging study. Will Flavahan led all of the work with the insulator loss in collaboration with Yotam Dreyer, who's now a faculty at Hebrew University, thanks to our collaborators at MIT, Penn, and of course at Dana Farber, George Dimitri, Praful Gokul, and colleagues as well. And I'm happy to pause now and see if there's some questions.
Brad, that was just fantastic. There are a number of questions. I'll try to sort of narrow them down, but feel free to scan through if you have that box open also.
Oh, yes, I see it now.
I guess one of the questions I would have is, you said that the TAD patterns are very disrupted in cell lines. So I was curious what you think is going on there, because to a certain extent, gene expression is very similar in the cell lines to sorted primary tumor cells. So how could there be that dissociation?
Good question. Let me clarify. The TAD boundaries are pretty well preserved in cell lines. It's the compartmental organization that's disrupted. So the cell lines still have the TAD boundaries. The problem is it's that higher organization of where the TADs are placed within the nucleus is totally disrupted. And that's what I was trying to convey. So, you know, I think it goes back to the stability of TADs, maybe explaining what you're pointing out, that the cell lines are actually decent surrogates on that level in terms of gene expression. But the more subtle effects of compartmentalization that I talked about in the first part of my talk are not well captured in the in vitro models, at least not the ones we tested. We want to test organoids and others, but not yet.
So then I guess to that point, when you say that TAD boundaries are fairly stable, that all these measurements represent to some extent averages across cell populations. And so how do you know within individual cells that they're truly stable, or that you're simply measuring compensatory averages?
Yeah, I'm glad you asked that too, because, you know, the reality is the field is moving really fast, and, you know, Xiaowei Zhang and, you know, really superb folks in our community are now imaging these kinds of structures at the single cell level, and they're hyperdynamic populations and they're ensembles, and we're still understanding what a TAD really means. A TAD does not mean that in an individual cell you've got this stable structure. It probably means that if you take these highly dynamic ensemble of cells, there's some tendency to stabilize or exist for just a little bit longer in organized by TAD. How that has such a profound effect on transcriptional activity, we're still trying to understand. But I think the best thing I could say is we've still got a lot to learn about how these behave at a single cell level. So I'm glad you asked that question. How did that happen? Did you hear me? I think I got muted.
Yep, we could hear that answer.
Oh, good. You know, we haven't looked at this extensively in leukemias. I think it's a really interesting question. Is it 3D? Oh, right.
So Brad, have you looked for mutations in that FGF insulator?
Oh, great question. I'm so glad you asked that. Muted. I keep getting muted. I don't know why, but I'm really glad you asked that question. So most GISTs are caused by KIT mutations. 15% of them seem to be caused by SDH loss, which we think causes the epigenetic activation of FGF. There's a really rare subset that was just published from a group that said there's some really rare GISTs that are not KIT and they're not SDH. We found that they have an FGF locus amplification. So yes, I think there is, you know, even rarer, I mean, like probably less than 1% of GISTs have amplified the FGF locus. So I think it supports the whole model that FGF is a really important driver, albeit in a relatively smaller subset of these tumors.
And there's a question, have you seen examples where there's a gain of insulator sites through this epigenetic mechanism?
Yeah, I've been really interested in that. I thought maybe these hypomethylated blocks would allow insulators to come in and enact them. I haven't seen much of that. I suspect that there will be because you see all these examples, but we haven't yet seen this. Somebody's asking about the relationship between what's upstream in terms of transcription and topological organization, and I would say that's also a rather hot area that folks are trying to figure out right now. I think that the sort of overriding messages I gave you about large-scale block hypomethylation and compartment shifts are driven by the methylation changes and the DNMT changes, and the consequences is transcriptional changes. I think that the example I gave you with the specific insulators being knocked out, the topology change is driving a change in expression. That said, there are also examples in other cases where a big change in transcription or can actually modulate boundary functions and so on. So yes, this can be bidirectional.
And then maybe I'll just ask one last question, and that is, when we make comparisons between tumor populations and normal populations, say in the colon, we use fully differentiated normal colon cells. But is it possible that the better comparator would be a less differentiated cell that might actually have topological and compartmental organization that's different from that fully differentiated comparator?
Yes, I think that's right. So can we get our hands on some tumor stem cells? Can we isolate tumor stem cells? To ask that question. I think the imaging approaches are going to allow us to begin to do some of that. Good question, and yes, we are setting up to look at some of that.
Well, I want to thank you again for just a spectacular talk, extremely well attended again. And as I mentioned, we'll break. We won't have a lecture tomorrow, but we're very fortunate to have Kathy Wu, who will speak on cutting edge aspects of cancer vaccination on Thursday. So thank you all for attending, and everyone, please be well.
Thank you for organizing, and thanks Nellie too. This is a great thing you're doing.